the most abundant form of the circulatory system is vitamin D3, and vitamin D3 undergoes when exposed to or is produced from the exposure of 7-dehydrocholesterol. When exposed to UV light, will break down into vitamin D3. That cholecalciferol will then be broken down in the liver and the kidney to your 125 dihydroxy vitamin D3. So we're naturally going to generate vitamin D3 with sunlight exposure. But if you live in an environment where you're not getting much sunlight exposure, then supplementation with vitamin D3 is a really good idea. So rhodopsin is the best understood role of vitamin A is its participation in the visual cycle in rod cells. The active molecule is retinal. So that is a vitamin A aldehyde. And the retinal forms an imine with the NH2 group of the protein opsin to form the visual pigment called rhodopsin. The primary chemical event of vision in rod cells is the absorption of light by rhodopsin, followed by isomerization to the 11-cis double bond to the 11-trans double bond. So this is not something, in fact, basically this and the next few slides I'm going to kind of touch on, but I'm going to migrate from them in favor of the discussion of membranes. So vitamin E is a group of compounds of similar structure. The most active is your alpha tocopherol. And what this is important, or what I want you to notice about this, is this chain right here. So this chain is made up of, it's a hydrocarbon chain, and what you'll notice is 1, 2, and 3 methyl groups coming off of it. These methyl groups are indicators of a compound known as an isoprene unit, or basically an isoprene unit is multiple isoprene units assembled to give us these. Isoprenes are key building blocks for pigment molecules as well as cholesterol. If you go on to take metabolism, you'll learn about isoprene units in cholesterol biosynthesis. And I also want to bring that up here. It's a little bit better visualized here, here, here. So here's those isoprene units. These are kind of things that can be repeated over and over again. What they're going to do is they're going to give a molecule an ability to move electrons around and absorb light. So at any rate, those isoprene units are big components of lipids and your vitamins. Now, changing gears to a discussion of membranes. Membranes are structures with many cellular functions. So first and foremost, I think the most obvious thing that a membrane does is it serves as a barrier to a toxic chemical or to toxic molecules. They help accumulate nutrients on the interior. They carry out energy transduction. They facilitate cell motion. They assist in reproduction, modulate cell signal transduction, and mediate cell-to-cell interactions. Your major membrane structures are displayed at the bottom: a micelle, a bilayer, and a liposome. Now, a micelle, the key thing about a micelle, imagine this is a sphere. The interior of a micelle is, well, these are hydrocarbon side chains. So very non-polar. So the interior, non-polar. Interior. A bilayer, well, this is just a cross-section of a bilayer, but a bilayer is going to be basically what makes up your cell membrane. And I've always kind of thought that a bilayer versus a liposome, a bilayer is a part of a liposome. And when you think about a cell as a whole or membrane-bound organelle, it's a bilayer. And the interior of it is similar to the interior of a liposome. It's an aqueous polar environment. But of course, within a bilayer, So right here, within that lipid bilayer, what do you have? You've got, well, a polar or a non-polar region. But on the outside here and here respectively, you've got polar regions. So those are your three major components or three major different cell membrane structures or membrane structures. Lipids are mobile in that they can move laterally very quickly. But if you take a glycerophospholipid and you want it to go onto the other side of the cell, that's going to be very slow. Now, with that in mind, lipids also have, just when we talked about fatty acids, melting point was a big thing when it came to the the length of a fatty acid determine the melting point of a fatty acid. Well, we've got that same sort of thing going on when we talk about the fluidity of a bilayer. So when a lipid is below the transition temperature, the bilayer becomes a very gel-like solid. Above the transition temperature, the bilayer is known as a liquid crystal. So the transition temperature is dependent on the chain length and the degree of saturation of its component fatty acid residues. So if you had, so I'll get into this in just a second with a study, but essentially at a low temperature, lipids are going to be very rigid. But then as you add heat, they get a little bit more fluid. They get a little bit closer to being a liquid as opposed to a solid. And it all has to do with this transition temperature. Now, cholesterol is going to broaden the temperature range of that phase transition. So cholesterol is going to influence membrane stability and fluidity. Now, here is a study that I think, if you take nothing else from our discussion of membranes, take this right here. What this was a table from a study in which E. coli was grown at 10 degrees, 20 degrees, 30 degrees, and 40 degrees. So at a very low temperature all the way up to a particularly high temperature. Now, naturally occurring E. coli is going to really love to be at about 37 degrees Celsius. So that's where it likes to be. But it can grow at 10 degrees, 20, 30, and 40 degrees Celsius. The rate at which it's going to grow is going to be a little bit variable. So at a lower temperature, it's going to grow very slowly. And whenever I'm saying grow, I mean duplicate. I mean, one cell becomes two at a very low temperature, very slowly. At 37 degrees, E. coli is going to go from one cell to two cells in about 30 minutes, whereas at 10 degrees, it might take, 6 hours or something like that. So the researchers for this paper sought to understand, well, what's the composition of the fatty acids in the bilayers? What's the ratio of what fatty acids are present, and what do we learn about them? And what they found was that at 10 degrees Celsius, that's where I want to start out, the percentage of fatty acids that were 14 and 16 carbons long, respectively, was about 22%. So less than 1/4 of the fatty acids were saturated fatty acids, about 14 and 16 carbons long. Now, at 10 degrees Celsius, the percentage of unsaturated fatty acids, well, 28 or 26 and 38. So we're talking about 64% of their fatty acids were saturated at 10 degrees Celsius. Now, what do we know about melting point and double bonds and saturation versus unsaturated fatty acids? We know that number of carbons causes the melting point, or as the number of carbons goes up, the melting point goes up. Number of double bonds goes up, your melting point goes down. So I've always kind of thought about this as we have some fluidity within the membrane. Now, if this was at 0 degrees Celsius, there would be basically no fluid. But what this shows is that the cells want to have a higher percentage of fatty acids that are saturated so that they can retain some mobility. Now let's go to the opposite end of the spectrum, 40 degrees Celsius. 56% of the fatty acids were saturated at 40 degrees Celsius. 21% were unsaturated at 40 degrees Celsius. So more than half were saturated and less than a quarter were unsaturated. So that is, you know, the numbers are a little bit off. But that's almost the saturation and unsaturation flipped at 40 degrees from what it was at 10 degrees Celsius. So we favored saturated fatty acids at 40 degrees Celsius, whereas at 10 degrees, we favored unsaturated fatty acids. So the way that I kind of process this is that if the temperature gets too high, ultimately, the membrane is going to lose all of its structural integrity. It's going to become too fluid. What a cell wants is it wants the membranes to remain intact so that it can keep the outside out and keep the inside in. It also wants to be able to have some mobility so that it can move around. Now, what we see here is we see a high percentage of monounsaturated fatty acids at a low temperature. So I imagine that if you look at my arms, imagine that's the lipid bilayer. This is the level of movement that we wanted. And you know what? The cell wants that same level of movement at a higher temperature. If you kept, or if there were 64% of the fatty acids were unsaturated at 40 degrees Celsius, then the mobility is like this. There's way too much motion. The structural integrity of that membrane is going to be compromised at that high of a temperature combined with that high a percentage of unsaturated fatty acids. But what we're able to do is find this balance where there's mobility, but not too much where everything's lost. And that is done by, at low temperatures, at low temp, favoring unsaturated. And at high temp, switching what we favor to the saturated fatty acids. Okay, so that I think is maybe one of the most important things to understand about this. You want, at all temperatures, the mobility of the cell membrane to be comparable. When you get to an extreme, however, when you get it to 100 degrees Celsius, there's going to be no structural integrity of that membrane. So that's a lost cause. But there's a range of temperatures in which that cell can continue to grow, that organism can grow. And that's going to be done by balancing the specific amounts of different saturated or unsaturated fatty acids. Now, phospholipids are synthesized on or near the interior of the membrane. There are two mechanisms to get the phospholipids from one side of the membrane to the other side of the membrane, because you have to have a phospholipid on the outside so that any adjacent cell can come in contact with that and communicate something to the cell that originates that phospholipid originates from. Flipases are a class of enzymes that catalyze the flip-flops of specific phospholipids in what is known as facilitated diffusion. Now, the uncatalyzed, if we look at these models over here, are uncatalyzed transverse flip-flop diffusion, movement of a phospholipid from one side to the other side. It's going to take about a couple of days. Whereas we can use an enzyme that moves this from the inside to the outside of the cell. And so that's going to happen very quickly in a matter of seconds. Whereas this movement from left to right on the same side of the membrane, that's uncatalyzed lateral diffusion, that's going to happen in microseconds. Now, so flipases, those are the class of enzymes that do this. That's what I want you to know. Now, the composition of a membrane. We've talked about phospholipids with their head groups. We've talked about cholesterol. But what I also want you to know about and be familiar with is some of the other components that are within a membrane. Now, they are also going to contain proteins. The proteins catalyze reactions, mediate flux of nutrient and waste, and participate in relaying external information to the cell. Protein to lipid ratios vary with membrane function. For instance, a human myelin sheath cell, 30% of the weight is protein, 30% of the weight of the membrane is phospholipids, 20% almost is cholesterol. If you take a yeast cell membrane, 52% is protein, 7% phospholipid, 4% sterol. If you take E. coli cells, 75% protein, 25% phospholipid, and 0% sterol, which is not too surprising because E. coli does not synthesize sterols. Now, the assembly, the organization of these proteins and other components. This phospholipid bilayer is referred to often as a fluid matrix. The bilayer is a two-dimensional solvent. Now, anytime you're shown a picture of a lipid bilayer, it's important to distinguish what's outside and what's inside of the cell. What's on the outside, what is displayed to the outside? Well, you've got these little hot dog looking things over here. Those would be your sterols. So sterols are actually found on the interior and exterior of the cell. Glycolipids are exclusively on the exterior. Oligosaccharide chains of glycoproteins are also exclusively on the outside. These are ways for mediating communication between cells, cell-to-cell contacts. Now, in the interior of the cell, there's quite a bit more. So we've got our peripheral proteins, where one termini of a protein or one part of a protein is only on the inside. Then we've got an integral protein with a single transmembrane helix. So that single transmembrane, we're going to have the anterior or N termini of a cell and the C termini of a cell, as an example, on opposite sides of the cell. That could also be the N versus the C. But the point here is it's a single transmembrane. There are peripheral proteins that are covalently linked to lipids. Those are going to be found on the interior of the cell. So if a protein has a lipid linkage, that protein is going to be found on the interior of the lipid bilayer. There are also integral proteins that have multiple helices, multiple transmembrane helices, in which case they might have a termini, both of their termini on the same side of the cell. Now, lipids and proteins can undergo rotational lateral movement, and membranes are asymmetric. So the stuff that's on the outside is going to be different from the stuff on the inside. Here's another display of basically that same information, just with space filling. Integral proteins are tightly associated, and in order to get them out of this membrane, you're going to have to use a detergent and or a chaotropic agent, which we've talked about chaotropic agents previously, something like urea, to disrupt that. So just to display a couple of examples, and I believe this is, transmembrane proteins that have a helix. So there's a couple of different classifications, and I want you to be familiar with these, but also I want you to be familiar with a figure like this. Now I'm going to start with this figure on the right-hand side. What this shows is a polypeptide, N termini to the C termini, and it shows a lipid bilayer. It shows the interior of that lipid bilayer, which we know to be extremely nonpolar. Now, what this also shows is amino acids that are within this lipid bilayer. We've got tyrosine, serine, isoleucine, leucine, isoleucine, threonine, glycine, isoleucine, valine, glycine, alanine, thionine, valine, glycine, isoleucine. Well, the proteins that are in this polypeptide that are embedded within this membrane in this very nonpolar region mostly are non-polar residues. Now, it's pretty common that if you look at the primary structure of a protein and you find a very densely packed region of non-polar amino acids, there might be some association with a lipid bilayer. That might be a transmembrane protein. So if you know the primary sequence of a protein, you can speculate, aha, that's going to, like, that region is very nonpolar. So maybe that entire protein associates with a lipid bilayer. Now, if we could look at these different types, well, I want you to be familiar with these different types and the organization of the terminal. We look at a type 1 protein, what do we see? Well, look at the bottom, we've got inside and outside. A type 1 transmembrane protein has a C termini on the interior of the membrane, interior of the lipid bilayer, on the internal side of the lipid bilayer. Whereas type 2 has the N termini on the inside and the C termini on the outside. Type 4, well, this is a protein that's pretty complicated, but what's worth noting is it's got only 1 and 2 termini. So this is one single polypeptide that has 1, 2, 3, 4, 5 transmembrane domains, 5 helices that span the membrane. Could it have more and still be a type 3? Absolutely. What this is really showing is that a type 3 has multiple helical TM domains. Now, type 3 and type 4 are different from one another because type 4 has a quaternary structure because it is multiple independent polypeptides that have folded together to make a large transmembrane domain. So this type 4 has multiple polypeptides. Each one of those polypeptides is a transmembrane domain. They come together to form something similar to a type 3. The difference is, though, is it one or multiple polypeptides. A type 5 protein has a lipid linkage that is embedded within the lipid bilayer. Type 4 is similar to a type 1 or type 6. I'm sorry. Type 6 is similar to a type 1 or type 2, but the difference is it also has a lipid linkage that associates with a membrane. So that's kind of the rundown of all these different types of proteins. I want you to know these different ones so that you'll know, is it a type 5? Where is it going to be? Is it, does it span the membrane, or is it associated and localized on one side of the membrane? Lipid linkages. Well, lipid linkages, some proteins are going to be associated with membranes through covalent attachments to lipids that anchor that protein to the membrane. These modifications can be farnesyl or geranyl residues, in which case it's 10 or 15 or 20 carbons long. I want you to understand that lipids are one way in which proteins can be linked to a protein, or linked to a membrane. Illustrated here are a couple of different types of ways in which a protein can be lipid linked. For instance, a palmitoyl group can be placed on a cysteine or a serine residue, and this palmitoyl group it's a fatty acid chain, it will insert itself into the membrane, and boom, when that palmitoyl group is on that cysteine, that protein that has that cysteine will now be embedded within the membrane. The N-myristoyl group. Well, there can be a myristic acid added to a glycine, or a myristoyl group added to a glycine that associates with a membrane. A farnesyl group can be added to a carboxy terminal cysteine. A cysteine residue embeds into the a farnesyl group embeds into the lipid bilayer. And then finally, this last one, this fourth one at the bottom, are GPI linkages, which are much more complicated. And these are linkages that associate a protein with a membrane on the outside. So I want you to know the amino acids: serine, glycine, cysteine, and the corresponding group: palmitoyl, myristoyl, and farnesyl. How proteins can be associated with a membrane on the interior. Now, this is just kind of the tip of the iceberg, but these associations are really important because of the fact that these associations get proteins where they need to be in some instances. If a protein is associated with a membrane and has a function related to, and location is important, well, if it doesn't get that lipid linkage, that protein doesn't really have a means to get to the location that it needs to be. If it doesn't get to where it needs to be, it can't catalyze the reaction that it needs to catalyze. So these lipid linkages are very important, and it's very important to understand these because they help deliver a protein where it needs to be. I believe that there's this aging disorder known as progeria, and progeria has been implicated as being the result of a lipid linkage that doesn't take place. And so what ends up happening is basically the cell cycle begins going at a very rapid rate, and someone afflicted with progeria can appear to be something like 80 years old when they're like 13. And as a result, they are unlikely to live beyond 13 or 15 or so. But anyway, these are very important for understanding different diseases. And that's where we're going to conclude our discussion of lipids. And I hope that this all helped. All right, have a good one.